evolution and ecology of the North American mountain goat. Let me tell you a little bit about it.
Mountain goats are known for their exceptional climbing ability and distinct appearance. From their thick white coat and muscular forequarters to padded mobile hoofs, mountain goats are adapted to the harsh environment they live in. Native populations are located across northwestern North America in alpine areas of the Rocky Mountains and Coast Range. The Northern Boreal Mountain Range contains close to 30% of the global mountain goat population. With the changing environment, we're witnessing species expansion south and to higher altitudes. As northern alpine specialists, mountain goats are especially vulnerable to climate change. They also face pressures from industrial development, recreational activities, and harvesting. The main goal of my project is to quantify the population structure and dynamics of the mountain goat across the range using genomic techniques.
Tissue samples from harvested animals are received in tubes like this and stored in our -80 freezer.
There are multiple ways to examine the genetic variation in the population. Sequencing whole genomes of multiple individuals provides the most information, but it is still prohibitive in cost for most studies. As an alternative, a number of reduced representation approaches have been developed that sequence only parts of the genome but still capture individual variation. A method that I'm using in my project is called RADseq, or restriction site-associated DNA sequencing. Preparation of a genomic library with this method spends almost a week. Once the DNA has been extracted from the tissue, the DNA strands are then cut at random sites with digestion enzymes. After, each sample is labeled with a unique barcode by attaching a short DNA sequence to the fragments. There are a number of quality control procedures in the process, such as visualizing each sample on an agarose gel. DNA fragments of a certain size are also physically selected for sequencing, as can be seen in these UV gel photos.
And here's the final library. In the small tube, I have the barcoded DNA fragments of 96 individuals that can now be submitted for next-generation sequencing.
From the sequencing data, we can identify genetically diverse subpopulations and estimate the inbreeding and migration rates. This fellowship has allowed me to increase my study to 400 individuals. With a larger sample size, those parameters can be resolved at a finer scale, making them more relevant for management and conservation strategies for the North American mountain goat.
I'd like to thank the W. Garfield Weston Foundation and WCS Canada for their generous support in making